Hello students, I hope all of you are doing good students. Welcome back to Vedanta Science Olympiad. This is Baswaraj sir, your biology master students, master teacher. All of you, welcome back students, welcome back. Hello. Students, am I audible and visible to all of you? Students, quickly let me know in the chat right now if I'm perfectly audible and visible to all of you students. Hello students, hello everyone. Good evening everyone students, good evening. Right. Now, हमने पहले क्लास में ना students the first part, right? Am I audible and visible students? Perfect. In the first class we finished until the entire glycolysis. हमने glycolysis तक पूरा का पूरा कंप्लीट कर दिया था and we have understood how एक ग्लूकोज है ना, एक ग्लूकोज से how we get pyruvate. This entire thing students, this entire thing is called as glycolysis. The first step is over. Now what is the next step? In the next step we will be understanding एक पाइरुवेट अभी उसके पास तीन रास्ते हैं अभी इस पाइरुवेट के पास. This pyruvate under two conditions which are absence of oxygen. In the case of yeast, it undergoes alcoholic fermentation. The same pyruvate in the muscle cell, it undergoes lactic acid fermentation. Also in lactic acid fermentation. The third pathway here is the presence of oxygen. In the case of mitochondria, it undergoes aerobic respiration students, aerobic respiration. Okay, that is we get CO2 plus H2O plus energy. यहां पर अभी हमारे पास तीन तरीके हैं. The first two types are anaerobic respiration. Both are called as anaerobic respiration. क्यों? क्योंकि यहां पर ऑक्सीजन की जरूरत नहीं है यहां पर और ऑक्सीजन इज नॉट इन्वॉल्व. The third category is called as aerobic respiration. Clear all of you students? Clear? Now, in the first two categories, first one we get alcoholic fermentation, second is lactic acid fermentation. Now students, now, do you want to understand the process in detail or do you want just the outline students? In the chat quickly. Do you want in detail how you want to understand it or do you want outline? All of you. Students, quick. All of you. Do you want the outline or do you want the detailed version? Both the fermentations, both alcoholic and lactic acid fermentation. Detailed version? Okay. So I want all of you to pay attention because you will not understand if you just not listen properly. Okay. Now students, let's start with the first one. First, we'll start with the lactic acid fermentation. Shall we students? Shall we? Okay. Now, first things first, which you need to understand here. In lactic acid fermentation, it happens without oxygen. यहाँ पर oxygen is not involved. Second thing is it happens in your lactic acid bacteria or muscle cells. Now, please understand very carefully, students. Okay. First things first. Now, here we have something called as NADH. Can anyone tell me where is this NADH coming from? Do all of you remember in the sixth step? In the sixth step of your glycolysis, there was something called as oxidation. याद है students? याद है सबको? In the sixth step of your glycolysis, there was oxidation. And from that sixth step, we got something called as NADH. याद है students? याद है क्या ये वाला step? अगर याद नहीं है, let me just show you once. Okay, friends, please listen to me very carefully. This particular NADH right here, this particular NADH right now undergoes something called as reoxidation. What happens? It undergoes something called as reoxidation. Now, if it undergoes reoxidation, what happens? Can anyone tell me what is, students, is there a lag? All of you in the chat quickly. If there is oxidation, is there loss of electrons or loss of protons or gain of electrons or gain of protons? Students, quickly in the chat. What is oxidation? Oxidation is in general loss, right? Loss of electrons and loss of protons. Now this NADH right here, it will lose electrons and lose protons. Electrons and protons are lost here. Now these electrons and these protons are accepted. The first electron acceptor in the case of lactic acid fermentation is pyruvate. This pyruvate is accepting the electrons and accepting the protons. Now, can anyone tell me this pyruvate right now is gaining electrons and gaining protons? So what will pyruvate technically undergo? Anyone in the chat quickly? This pyruvate right now undergoes something called as reduction. It undergoes something called as reduction by gaining the electrons and protons from here, and as a result of reduction we get something called as lactate here. Lactate. Now 3 carbon 3 carbon, it means there is no decarboxylation happening here. Okay, and the enzyme involved in this entire process is called as lactate dehydrogenase. How many of you understood? See, I could have simply told you pyruvate to lactate, that's all, enzyme, but I made you realize what is the depth concept here. Very simple concept, that is NADH undergoes reoxidation and electrons and protons are released. Those are taken up by pyruvate that undergoes reduction to give lactate. How many of you understood? Very simple, बहुत ही simple है. All of you understood? Done. Now, same thing happens in the case of alcoholic fermentation. Same thing. But here, there is a question. There was a PYQ, okay, in AIMS PYQ, okay. There was a, so, yes, okay. The acid mainly has the acidic component, more H plus ions. That's all, okay. Now, can anyone tell me the first electron acceptor in the case of alcoholic fermentation? They can ask you. Now here there's a small difference here. यहाँ पर क्या हो रहा है पता है? This pyruvate is directly not converted to ethanol, not converted. अब क्या होगा पता है? This pyruvate will lose out a carbon dioxide. That is basically decarboxylation is happening here. Decarboxylation is happening. That, so from this pyruvate 3 carbon we get a 2 carbon compound called as acetaldehyde. Decarboxylation. Done? Done. Now after this, it's the same process again, same process. What happens? This NADH undergoes what, students? This NADH undergoes reoxidation, and after reoxidation this NADH will lose electrons and protons here. These electrons and protons are taken up by acetaldehyde right now. Acetaldehyde. So acetaldehyde is technically the first electron acceptor in the case of the alcoholic fermentation, students. Alcoholic fermentation. Okay. Students, is my voice proper or is it breaking? Students, quickly in the chat. Is my voice proper or is it breaking, students? Sound check, sound check. No. Now this acetaldehyde is gaining electrons. So this acetaldehyde is gaining electrons. It undergoes something called as reduction right now. Now, in the case of alcoholic fermentation, we get ethanol, which is 2 carbon compound. So is it breaking or proper? Breaking right now? Students, it is there in NCERT, but I'm teaching you properly. Like NCERT, if you read, you'll understand. But if you understand this part here, right, you'll understand even better. Okay, let me just show you, students, if you don't believe me. Let me just show you NCERT, students, NCERT. Respiration implants, NCERT PDF. Many students just skip this, but we learn in detail, right? Okay. See this, students. All of you can notice here? Can all of you notice here? Okay. See here, in animal cells also, like muscles, right? Muscle, like muscles during exercise, when oxygen is inadequate, इसका मतलब क्या है? इसका मतलब है anaerobic respiration. First respiration, pyruvic acid is reduced. Can you see, students? Pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH, which is reoxidized to NAD plus in the both the process. Students, you understand? So it is given in NCERT, but you don't understand very well if you don't learn it this way. So NADH is the reducing agent. It is undergoing reoxidation to release electrons and protons, and in alcoholic fermentation acetaldehyde undergoes reduction. In the case of lactic acid fermentation, pyruvate undergoes reduction. Easy? Very easy, right? Now students, now let's say they ask you a question, how much ATP is produced in anaerobic respiration? Anyone, what is the answer? Anyone knows the answer? how much ATP is produced in the case of your alcoholic fermentation or lactic acid fermentation? कितना ATP बनता है यहां पर? Anyone? Okay. Now Preeti is saying 2, Divanshu is saying 2, but how is it 2? Do you want to understand, students? Do you want to understand how is it 2? Do you want to understand how is it only 2 molecules of ATP? Do you want to understand? Okay. Very simple logic, very simple logic. Now can anyone tell me, in the case of your preparatory stage, how much ATP is lost? Glycolysis. In the case of glycolysis, preparatory stage, minus 2 ATP, है ना? Minus 2 ATP. Now in the payoff stage, what happens? We generally have the NADH, which we have 2 NADH and 1 NADH gives you 3 ATP. So 2 into 3 is, यहाँ पर generally 6 ATP बनता था यहाँ पर. Generally 6 ATP was formed here because of 2 NADH in glycolysis. इसको छोड़ के ना, इसको छोड़ के we had 4 ATP in the, how many, which step, can anyone remind me? Which step this 4 ATPs are formed? दो ATP कहां पर बनते थे स्टूडेंट्स? In the 7th step and the 10th step. 7th step और 10th step में ना दो ATP बनते थे, जो 4 ATP टोटल थे ना, 4 ATP थे. वेरी गुड. सो यहां पर कितने ATP बने? 6 plus 4 10 ATP. 10 is the gross. 10 minus 2 8 ATP बनता था नॉर्मल कंडीशन में. नॉर्मल कंडीशन में ना हमारे पास 8 ATP बनते थे ना. अभी हम क्या पढ़ रहे हैं स्टूडेंट्स? हम यहां पर पढ़ रहे हैं कि अब आराम से देखो स्टूडेंट्स, आराम से देखो. यहां पर देखो, दिस इज ग्लाइकोलिसिस, ग्लिसरॉल्डिहाइड 3 फॉस्फेट. यहां पर एक 6 स्टेप में NADH बन गया यहां पर विद द हेल्प ऑफ ऑक्सीडेशन. विद द हेल्प ऑफ ऑक्सीडेशन यहां पर बन गया यहां पर. उसके बाद क्या हो रहा था? इस पायरुविक एसिड, इट इज अंडरगोइंग लैक्टिक एसिड फर्मेंटेशन हियर एंड अल्कोहलिक फर्मेंटेशन हियर. इन दोनों प्रोसेस में ना, इन दोनों के दोनों प्रोसेस में ये जो NADH हमारा बन के आ रहा था ना, ये एक्चुअली यहां पर यूज हो रहा है. ये क्या हो रहा है स्टूडेंट्स? यहां पर यूज हो गया अभी. तो ये NADH रीऑक्सीडाइज होके यूज हो गया अभी. So can we consider this here? All of you, can we consider? We cannot. So we are not considering this entire ATP generation at all. So if you are not considering the 6 ATP, so 4 minus 2 is how much students? 4 minus 2 is how much? 2 ATP. So हमने बताया था ना, पहले क्लास में बताया था कि यहां पर सिर्फ वेरी लो ATP बनता है ना? वेरी लो ATP बनता है यहां पर, वेरी लो ATP बनता है. और ये वेरी लो कैसा है? 2 ATP है यहां पर. स्टूडेंट्स इजी है ना? इजी है ना? बहुत इजी है. ओके स्टूडेंट्स, कैन वी गो टू नेक्स्ट कॉन्सेप्ट? यस, कैन वी गो टू नेक्स्ट कॉन्सेप्ट नाउ? सो ये नहीं बना भी यहां पर. सो 4 minus 2 is how much? 2 ATP. yes, okay, listen to me, I'll explain one more time, don't worry, आराम से समझो. नॉर्मल कंडीशन में स्टूडेंट्स, नॉर्मल कंडीशन में कितने ATP बनते हैं? नॉर्मल कंडीशन में 8 ATP बनते हैं यहां पर, है ना? इतना याद है सबको? इतना याद है. ओके अभी ये क्या हो रहा है पता है? इस अल्कोहलिक फर्मेंटेशन या लैक्टिक एसिड फर्मेंटेशन है ना, इस दोनों के अंदर ये 2 ATP एक्चुअली यूज हो रहे हैं, क्योंकि 2 टाइम हो रहा है प्रोसेस. सो अगर ये दोनों NADH यूज हो गए, तो इसका मतलब क्या है? जो हम कैलकुलेशन में उठा रहे थे ना, पूरे कैलकुलेशन में ये 6 ATP हम काउंट नहीं करेंगे, अभी इसको काउंट नहीं करेंगे. तो इसका मतलब क्या है? अभी हमारे पास क्या है? हमारे पास ये 4 ATP यहां पर है और ये 2 ATP यहां पर. 4 ATP यहां पर और ये 2 minus 2 ATP. So what is 4 minus 2? 4 minus 2 is what? 2 ATP. 2 ATP बन गए, उतना ही. So very low ATP. Students, my dog name is Cash. His name is Cash. His name is Cash. बाहर पटाखे फोड़ रहे हैं ना, तो ये थोड़ा पागल हो रहा है. Okay. So in the class, today's class, you will understand. You will hear his sound very much. Okay. क्योंकि बाहर पटाखे फोड़ रहे हैं, obviously. Okay. Now students, now let's start with aerobic respiration, shall we? Yes. Sir, why NAD is 3 ATP, students? It is actually not 3 ATP. It is actually 2.5 ATP. Okay, because you'll understand how it is 2.5 ATP because of the ETC. जो NADH और FADH यहाँ पर बनते हैं ना, this NADH and FADH is sent for ETC. What is ETC? Electron transport chain. इलेक्ट्रॉन ट्रांसपोर्ट चेन में ये हाई एनर्जी मॉलिक्यूल्स ATP प्रोड्यूस करने में हेल्प कर रहे हैं. किस तरह से? because this NADH and FADH are donating H plus there. They're donating H plus, और ये H plus आगे जाके ATP बनाता है. Okay, I'll teach you, don't worry. बहुत easy है. Okay, okay. देखो, बट अगर आपने एग्जाम में पूछेंगे तो एग्जाम के अंदर जस्ट 3 और 2 याद रखो काफी है. 1 FADH gives 2 ATP, 1 NADH gives you 3 ATP. पर एक्चुअल इंफॉर्मेशन क्या है? एक्चुअल इंफॉर्मेशन है 2.5 1.5. ओके. हाँ, यस दे कैन टेक्निकली, बिकॉज इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम, इलेक्ट्रॉन ट्रांसपोर्ट चेन एक ही चीज है ना. लाइक दे आर टॉकिंग अबाउट द जंपिंग ऑफ इलेक्ट्रॉन्स. वहां पूछ समझ रहे हैं वहां पर एक सिस्टम. व्हाट इज स्टूडेंट्स, आई हैव टोल्ड यू ऑलवेज, व्हाट इज द मीनिंग ऑफ सिस्टम? यस, व्हाट इज द मीनिंग ऑफ सिस्टम? मतलब क्या है? सिस्टम का मतलब है कि मल्टीपल कंपोनेंट्स आर वर्किंग टुगेदर फॉर वन पर्पस. सोलार सिस्टम है ना, सोलार सिस्टम, कंप्यूटर सिस्टम, digestive system, electron transport system, same meaning. Okay. So why NCERT printing wrong? Because students, NCERT is little cuckoo in the head. तो उसको दिमाग कभी कभी चलता नहीं है. Okay. Yes. Students, bio is very vast. 2 point मनोज 2 और 3 और 2 याद रखो काफी है. 2.5 0.5 नहीं चाहिए, 3 और 2 याद रखो काफी है. क्योंकि एनसीईआरटी को यूजुअली मानते हैं ना, तो एनसीईआरटी में 3 और 2 लिखा है, उतना ही. Okay. Now students, now let's do aerobic respiration, है ना. Now in students important topic, students आपको चाहिए एक आपको एक वीडियो चाहिए top 50 questions of, I will have to spend around 50 hours I feel. Okay, top 50 questions from biology for NACJS examination. Students, do you want that? Yes, because I have to spend a lot of time on it, so I'll sit a lot of right. So so I will sit, I will say, I think I have to spend 2 days because I can't give you wrong information. Okay, so I have to I have already solved a lot of PYQs. So अब क्या करेंगे पता है, हम एक शायद से 2-3 दिन लगेंगे, उसके बाद एक वीडियो आएगा top 50 questions from biology. Please make sure you know all 50 topics from there. Okay, so please make sure you know all the 50 topics from that video which I'll tell you. Okay, I'll do that, students. Okay, students, I'll bring marathon. We will do okay. Students, revision हो रहा है. Students, do you realize I also teach for K? Students, बीच में मैंने classes कम करी थी क्योंकि I was supposed to teach also second PUC. I teach for PU also, right? PU is 12th standard. Students, recent video didn't check, students. Okay, okay students, let's understand aerobic respiration. It's बहुत ही easy है, आराम से समझो, बहुत ही easy है. ठीक है, यहाँ पर क्या हो रहा है, ग्लूकोज प्लस ऑक्सीजन, एनर्जी रिलीज बिकॉज इट्स एरोबिक रेस्पिरेशन. इस एरोबिक रेस्पिरेशन में एनर्जी निकल के आता है, एंड वी हैव सर्टेन बाई प्रोडक्ट्स हियर सच एज योर कार्बन डाइऑक्साइड एंड वाटर. नाउ व्हेन इज दिस CO2 रिलीज्ड इन प्लांट्स? पानी कहां पर निकलता है? बोथ ऑफ देम यू हैव टू आइडेंटिफाई इन द एंड ऑफ द चैप्टर. ठीक है स्टूडेंट्स, आइडेंटिफाई करोगे ना? ओके, आइडेंटिफाई करोगे स्टूडेंट्स. Students, I will suggest you along with NEET PYQs, please do AIPMT and AIMS PYQs. Okay, please do AIPMT and AIMS PYQs along with NEET PYQs. NEET PYQs थोड़े easy होते हैं आपके exam point of view से. Okay. Now students, now can anyone remind me, glycolysis cytoplasm में हो रहा है ना? Glycolysis cytoplasm में हो रहा है. यहाँ से क्या बन के आया था? Pyruvate बन के आया था. Now this pyruvate will enter inside the mitochondria. This pyruvate will enter inside mitochondria, और इस mitochondria के अंदर ना 3 दूसरे reactions होते हैं. First one is your link reaction, then we have Krebs cycle, then we have ETC. 3 different reactions inside the mitochondria right now. Inside the mitochondria. Now, first one is your link reaction, which happens in the matrix, also called as oxydecarboxylation. Second happens in the mitochondrial matrix. Third happens in the inner membrane. Okay. Now let me ask all of you. Do you want to learn the detailed version of link reaction, or do you want the easy version? Easy version, I'll tell you what is the easy version. Easy version is pyruvate converting it to acetyl-CoA. That's all. Now students, let me ask you again. Do you want a detailed version? Yes? Hmm. 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 all of you. Yes, students, you know, right? Even if you say detailed, even if you say easy way, I'll teach you detailed way only. You don't have an option, you know, you don't have an option. Okay. Okay, students, now please focus for 2 minutes. 2 minutes focus, because if you don't focus here, you might lose on one point here. Okay, now this is called as link reaction. Now you might ask me, sir, Q, why do we call it as link reaction or gateway reaction? Gateway reaction क्योंकि पता है, this is a reaction which is happening between glycolysis and Krebs cycle. इन दोनों के बीच में होता है the link reaction. Okay, so यहां पर है glycolysis, इस तरफ है हमारा Krebs cycle, इन दोनों के बीच में है the link reaction. Now link reaction is also called as oxidative decarboxylation. क्यों, इसका मतलब क्या है, आराम समझो. Now can anyone tell me what is decarboxylation first? Anyone in the class? Anyone in the class who can tell me what is decarboxylation? Anyone? Decarboxylation मतलब removal of CO2 है ना, removal of CO2. So पहली बात यह है कि this is a pyruvate. This is a pyruvate from where? This is a pyruvate from the after glycolysis. Glycolysis के बाद the first step is there is removal of CO2. Removal of CO2 is happening right now. After you remove CO2, what do we get? After removal of CO2, we have what? COCH3 है ना, COCH3 है ना, COCH3. इसका मतलब क्या है? What is COCH, students? COCH3 is basically the acetyl group. Acetyl group we have right now. We have the acetyl group right now. Okay, acetyl group right now, acetyl group. Now what happens? Understand, students, understand. Let me just finish this before taking doubts. Okay, now we have the COCH3 here. Now this COCH3 undergoes something called as oxidation. Oxidative, right? Oxidative. So this COCH3 undergoes something called as oxidation. Now can anyone remind me what is oxidation? Can anyone remind me what is oxidation? Oxidation is removal of removal of electrons or loss of electrons and loss of protons. So what is happening is after decarboxylation, it undergoes oxidation. When acetyl group undergoes oxidation, it is losing electrons and protons. Electrons and protons are lost. Now this electrons and protons are picked up by a electron carrier, we will always call it. Okay, that is basically your NAD+. NAD+ is picking the electrons. Now students, can anyone guess what happens next? This NAD+ is picking electrons. So what is it undergoing? Obviously, if it is gaining electrons to become NADH, it undergoes reduction. So this NADH undergoes reduction to make NADH here. This pyruvate undergoes decarboxylation and oxidation to produce something called as acetyl plus. We also add something called as coenzyme here. So acetyl CoA. We get something called as acetyl CoA. Now students, in this entire reaction, we have something called as an enzyme involved here. Okay, a enzyme and a cofactor. Now before I tell you what is a cofactor and an enzyme, I believe all of you might know from biomolecules chapter. Can anyone tell me which is a protein and which is a non-protein part in an enzyme and a cofactor? It's a PYQ, all of you. All of you should be aware of this. Enzyme is generally a protein part. Cofactor is non-protein part. है ना? Cofactors are non-protein. All of you remember, right? Don't remember biomolecules. Yes. Remember or not? Yes. Apoenzyme. Enzyme. Yes. Protein part. Apoenzyme is all of you please remember. Okay, students. नहीं पढ़ाया. Okay, students. Okay, what we'll do is next tomorrow we'll do. Students, tomorrow we'll do. No, not tomorrow. Wait. कब था हमारा ये students? कल कर सकते हैं. Actually कल कर सकते हैं students. कल कर सकते हैं. रिवीजन इस वीक में करेंगे स्टूडेंट. 1 मिनट को एक काम करते हैं, कल या परसों, इन दोनों दिन में ना, कल या परसों में ना, एक दिन में वन शॉट बायोमोलिक्यूल्स रिवाइज करवा दूंगा एक बार, क्योंकि आई फील एवरीवन थिंक्स इट इज अ बैकलॉग, राइट? सो इन वन शॉट आई विल फिनिश इट. ओके. ओके. Sir, but enzyme is the catalyst. But students, please remember, enzyme is a protein. Enzymes are generally, apart from your ribozyme, enzymes are protein. Please remember, enzymes are protein. Enzymes are made up of proteins, right? And these protein molecules will have a lot of places where the cofactor will join with them, right? So please remember. Students, ecology is over. So animal kingdom is also over. Okay. Now students, let me just recap this once to clarifying. This link reaction is also called as oxidative decarboxylation. Why? It is called as oxidative decarboxylation because first step is decarboxylation. Pyruvate loses the CO2, undergoes decarboxylation to produce your acetyl group. Now this acetyl group undergoes oxidation. Once it undergoes oxidation, it will lose electrons and protons, lose electrons and lose protons. And these electrons and protons were taken up by your NADH, which undergoes reduction to get NADH here. Okay, NAD plus. Now, after oxidation, we add a coenzyme part here. That's why we get coacetyl-CoA. That's all. एक मिनट स्टूडेंट दिखाता हूं आपको मैं. ये देखो स्टूडेंट्स. Here. And in this entire reaction, we have an enzyme here. The enzyme is called as pyruvate dehydrogenase, and the cofactor involved is called as Mg2 plus. Students. Now all of you can understand how pyruvate, we add a coenzyme here, we have NAD plus. समझ आ रहा है? NAD plus. How we get acetyl CoA, है ना? Because of oxidation we get this, and because of decarboxylation यहाँ पर एक CO2 निकल के बाहर आ रहा है, and because of reduction we get NADH. Friends, all of you are understanding. It was, my job would have been very easy. मेरा job बहुत easy होता if I just showed you this reaction, है ना? I could have just showed you the reaction. देख लो एक बार दिखा दो. But it is important to understand how we get this acetyl CoA because of oxidation and decarboxylation reduction. Done? Done, students? Done? Yes? Okay, एक बार फिर आराम से students, आराम समझो. What happens first? What happens first is pyruvate undergoes what? Pyruvate undergoes decarboxylation. First point, decarboxylation, done. Now after what happens? After decarboxylation, we get acetyl group. है ना? We got acetyl group. Which is acetyl? After decarboxylation, we got acetyl group. Now this acetyl group undergoes what? Oxidation. Second step. Please remember. Now once it undergoes oxidation, this acetyl group will lose electrons and lose protons. This electron and this proton are taken by your NAD plus. So NAD plus will gain electrons, reduction to NADH. Now once it is undergoing oxidation, उसी वक्त we also add a coenzyme to get acetyl CoA. That's how is the reaction happening. That's how the reaction is happening. So we get this acetyl CoA because of oxidation, right? Oxidation. We get this CO2 because of decarboxylation. We get this NADH because of reduction. Students, the detailed version all of you asked for. Okay. Now, students, now. No. Done. Now, what do we have, students? Now, what do we have is we have the acetyl CoA part. And now we have the acetyl CoA, students. So, because people ask, see, students, I understand some of you learn very easily, some of you take time, but it's fine. Okay. I always make sure students understand. If it is one student also, I have explained, you will not believe me. In the paid batch, students asked me this 7 times. I explained 7 times this concept. Okay. Until everyone understood. Okay. Now, students, all of you know what is acetyl CoA right now. We have acetyl CoA right now. है ना? हमारे पास acetyl CoA है अभी. It's a 2-carbon molecule. All of you know it is a 2-carbon molecule. Why 2-carbon molecule? Because of decarboxylation. Pyruvate था 3 carbon, है ना? Pyruvate था 3 carbon, right? 3 carbon से 1 carbon निकल गया ना? So we have 2 carbon here. Acetyl CoA serves as a fuel for citric acid cycle. Link between glycolysis and Krebs cycle. Link reaction. Now, this is done, students. This is done. This is done, students. This is done. Now, next is Krebs cycle. Shall we start, students? Very easy Krebs cycle. Okay. Now, here I'll give you a small trick also to remember Krebs cycle, and along with that, I'll make sure you understand every single step of Krebs cycle. Friends, shall we start? Again, do you want a detailed version or do you want simple upar se? Right, very easily. Students, I play football every week. I will destroy all of you. I play football 3 times or 4 times a week. I will destroy all of you. I play for Bangalore B division team. Okay. So if you think you can win against me in football, joke. It is joke. Right. I am very sure all of you can't beat me in football. All of you can't beat me in playing games also, video games. Okay. I don't think so. You can beat me in cricket. I can't play, students. Cricket I can't play. I'm very sure all of you can't beat me in games also, because see here also this right here is my controller also. Okay, so very sure you can't beat me in games. Now, students, now let's have the Krebs cycle. बहुत easy है. The Krebs cycle was, why is it called a Krebs cycle? Krebs. The meaning the Krebs is because of the scientist who discovered the Krebs cycle, called as Hans Krebs. You know, this scientist called as Hans Krebs. Okay, Hans Krebs, students. Now, here we have the Krebs cycle, students. Now, do you want a trick, students? एक छोटा सा trick दूंगा. All of you will remember the Krebs cycle very well. Okay, the small trick. Okay, small trick. The small trick is very simple. Officer, can I keep selling? Officer, can I keep selling sandwiches for money? For money. Okay. Okay. So not crabs, crab. Okay. Officer, can I keep selling sandwiches for money? इसका मतलब क्या है? Understand, students. Understand. The O in the officer stands for oxaloacetate. The C stands for citrate. I for isocitrate. K for alpha ketoglutarate. S for succinyl coenzyme A. S for succinate. F for fumarate. M for malate. Students, और एक बार. O stands for oxaloacetate. C stands for citrate. I stands for isocitrate. K stands for alpha ketoglutarate. Stands for succinyl coenzyme A. S for succinate. F for fumarate. M for malate. That is your entire Krebs cycle. Now, in examination, they will never ask you to draw the Krebs cycle. नहीं पूछेंगे. This is not board exam. So what you need to know is the concept here. Okay. Or I'll tell you exactly what they'll ask you also, which they have been asking in the previous classes. Okay. So students, shall we do point by point right now? Students, start करें क्या? Point by point. Shall we start? Okay. Now the first point is, student, the first point is, here we have the 4-carbon oxaloacetate. 4-carbon oxaloacetate, students. How many carbon is acetyl-CoA? Acetyl-CoA, students, is what? Yes. Acetyl-CoA is what? How many carbon? 2 carbon. 2 carbon. So 2-carbon acetyl-CoA is entering the Krebs cycle. It is joining with oxaloacetate. So what do we get citrate as? How many carbon is citrate, students? How many carbon is citrate? Citrate right now is 6 carbon. 6 carbon citrate. First point clear? Very good. Okay. Now, students, understand what happens. It takes place in mitochondrial matrix. Very important. Where does the Krebs cycle takes place? Mitochondrial matrix में होता है ये वाला. Okay. Now what happens? Acetyl-CoA plus oxaloacetate, they undergo joining. When this joining is happening, remember this right? Remember this coenzyme here? This coenzyme A is actually removed outside. Why? Because this coenzyme A will again go join back in the link reaction. निकल जाएगा बाहर. And then we get this entire citrate here, students. Citrate here. So citrate is how many carbon? 6 carbon. उतने याद रखो काफी है. Now, students, now what happens? Okay. The first molecule in the Krebs cycle is what? The first molecule in the Krebs cycle is citrate. So can I also call it as citric acid cycle? All of you? So Krebs cycle is also called as what? Krebs cycle is also called as citric acid cycle. इतना याद रखो काफी है. citric acid cycle. Okay, now it is also called as what? It is also called as TCA cycle. कभी सुना है students? कभी सुना है? It is also called as TCA cycle. Why? It is called TCA cycle because of the structure of citrate. If you look at the structure of citrate, can anyone tell me, students, how many carboxylic acid groups you have here? Anyone? In this structure of citrate, all of you. Students, if you want, obviously you can. Right? You have to be very dedicated. You have to question solve करना है आपको. Okay. Tell me, students, how many carboxylic acid do you see here? Tell me, students, tell me quickly. How many do you see here? I see 1, 2, 3. Yes, 3 are there. So can I call it tricarboxylic acid cycle? So there are multiple names of your Krebs cycle. So please know all 3 names. है ना? It's called as Krebs cycle. It is called as TCA cycle. It is called as citric acid cycle. Clear? Perfect. Okay. Now, students. Now, this step, I don't think any doubt should be there. Now, do you see any decarboxylation here? 6 carbon. And citrate to isocitrate, there is only isomerization. Okay. 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 Isomerization. Normal isomerization is happening here. Okay. Isomerization is happening. Okay. Just change in the structure is happening. Okay. Now, students. Now this step and this step is very important. Okay. Okay. Okay, students. Okay. यहाँ पे क्या हुई है? In this step, I call it something called as OD. What do we call it? OD1 and OD2. OD1 and OD2. तो इसका मतलब क्या है स्टूडेंट्स? इसका मतलब क्या? What is OD? OD का मतलब है ऑक्सीडेटिव डीकार्बोक्सिलेशन हो रहा है यहाँ पर. So एग्जामिनेशन में यही पूछेंगे आपको. Which steps oxidative decarboxylation is happening? So between isocitrate to alpha keto, OD1 is happening. OD1 is happening here. And between alpha keto and succinyl coenzyme, OD2 is happening. But what is OD right now? OD का मतलब क्या है? आराम समझो students, आराम समझो. Okay. Now what is OD understand students? Okay. Isocitrate is converted to alpha. Isocitrate is converted to alpha ketoglutarate right now. The first thing which happens here, student, the first thing which happens here is decarboxylation. First thing is what? Decarboxylation. So this isocitrate से ना एक CO2 बाहर निकल के आएगा. Okay. So if the CO2 is lost here, obviously there is a reduction in the carbon number. यहाँ पर था कितना? 6 carbons. यहाँ पर कितना हो गया? 5 carbons. So let's write here only, student. Let's write here only. So यहाँ पर कितने carbons हैं, students? We'll go to the first slide here. Okay. So how many carbons in the alpha keto, students? Alpha ketone is the only 5 carbon molecule. है ना? यहाँ पर थे 6 carbon और यहाँ पर 5 carbon हो गया. So the meaning of OD. OD is what? Oxidative decarboxylation. Okay. Now, students. निकल के आ गया, निकल के आ गया. Now what is happening, students? Now what is happening is this entire isocitrate undergoes oxidation. It undergoes oxidation. Now if it undergoes oxidation, will it lose electrons and protons? All of you, yes or no? हाँ. So ये isocitrate अभी electrons and protons lose करेगा. Electrons and protons are lost. It are lost. So when these electrons and protons are lost, they are now picked up by NAD plus. NAD plus. And this NAD plus undergoes reduction right now to get NADH. To get NADH here. So that is the meaning of OD1. The meaning of what is OD1? The meaning of OD1 is first step is decarboxylation. The second step is what? Second step is oxidation. और ये जब ये oxidation होता है ना, these electrons and protons are taken up by NAD plus and converted to NADH. That's how we get NADH here and CO2 here. Now that is called as OD1, oxidative decarboxylation. जहां पर इस isocitrate से एक CO2 निकल के बाहर आ गया और उसके साथ this undergoes oxidation. and that's how this electrons and protons are lost, and that is picked up by NADH to get reduced to NADH. Now students, here the second OD is happening. Let's say second OD is happening. Can anyone tell me what is the carbon number of succinyl coenzyme A? Anyone? What is the carbon number of succinyl coenzyme A, students? Carbon number? All of you. Quickly, students, in the chat. Carbon number here. Second OD is happening here. What is the carbon number? Obviously, students, obviously. यहाँ पर थे 6 carbon. This was 6 carbon, right? Let's go back here. See, if you remember correctly, students, in the OD1, after OD1, we had 5 carbon. So after OD2, obviously, this succinyl coenzyme is 4 carbon compound right now. 4 carbon compound. Very simple, 4 carbon compound. Now again, what is happening? This alpha ketoglutarate undergoes decarboxylation. निकल क्या बाहर आ गया? Second step is what? Second step is this alpha ketoglutarate undergoes oxidation. है ना? This undergoes oxidation. Now once it undergoes oxidation, loss of electrons, loss of protons. Yes, yes, yes. So where is 24? What 24 are you talking about? Okay, okay. This alpha keto will lose electrons and protons after oxidation, and that is picked up by NAD plus to get NADH. NAD plus is converted to NADH here because of reduction. You know? That is what is happening. Now here, can all of you notice there is no more OD at all. No more OD at all after that. No more OD at all. Next, what they can ask you here is how many molecules of substrate level phosphorylation is happening in one Krebs cycle. Now, what is this substrate level? Right? What is this substrate level phosphorylation? The meaning of substrate level phosphorylation is how much of ATP is directly produced without any conversion of NADH or FADH, directly produced, and the answer is only 1. Can you see, students? This 1 GTP is produced in one cycle. One cycle, only 1 GTP is produced. एक ही GTP बनता है यहाँ पर. Okay, between succinyl coenzyme A to succinate, only 1 GTP is produced, and this is produced substrate level. Substrate level. Meaning of substrate level is using this, what is GTP? Guanosine triphosphate. है ना? ATP, GTP, CTP. पता है ना students? आपको पता नहीं? Okay, sorry students, my wrong, my bad. Okay, so GTP is guanine triphosphate. It converts to ATP later on, adenosine triphosphate. Okay, guanosine triphosphate. Students, energy, it's our energy molecules. GTP, ATP, है ना? Right? Okay, so students understand. This ATP is called as ATP because of, आपको पता है, there are 4 nitrogenous bases. आपको पता है ना students? All of you know there are 4 nitrogenous bases. A, T, G, C. पता है आपको? Purines and pyrimidines. Students, am I talking to myself? पता है ना students? हाँ. The only difference between ATP and GTP is the ring present there. Ring. In case of your ATP, adenine ring is present. GTP, guanosine ring is present. That's all the differences. Okay. Energy levels, they're same. Okay, energy levels are same. Okay. So ATP, GTP, TTP, CTP बनते हैं. ऐसे बनते हैं. Okay. Okay. So here we have GTP molecule. We get a GTP molecule. This GTP molecule will convert to ATP molecule later on. ATP molecule later on. Okay. This is for one cycle of 1 acetyl-CoA. Okay. Guanine ring. Yes, guanine ring is present. Okay. Now students, here we get succinate. Succinate to fumarate, we get one FADH2. This is produced as a result of normal oxidation. OD नहीं है यहाँ पर. Normal oxidation, we get FADH2. Then water molecule. In the last step again, we get, because of normal oxidation, we get one more NADH. Now exam में question कैसे आएंगे? आराम से question समझो, आंसर बताओ. आराम समझो. From one glucose, how many molecules of NADH I get in Krebs cycle? Students, आंसर बोर्ड के अंदर ही देखो, बोर्ड देखो आराम से, आंसर पता चलेगा. From one glucose, how many molecules of NADH do I get in one Krebs cycle? In one Krebs cycle, how many NADH do I get? How many NADH? I'm not talking about ATP, I'm just talking about NADH. How many do I get? All of you are wrong. All of you are wrong. What is the question? The question says from one glucose, one glucose, one glucose gives 2 pyruvate. One glucose gives 2 acetyl CoA. One glucose gives 2 acetyl CoA. 2 acetyl CoA. और एक acetyl CoA से 3 बनते हैं. तो 2 से कितने बनेंगे? 6 बनेंगे. Answer is 6. Answer is 6. Clear all of you? Clear right now? So this is for one acetyl CoA. One acetyl CoA, one cycle. But the question was what? The question was from one glucose. एक ग्लूकोज से कितने बनते हैं? कितने बनते हैं? 2 बनते हैं. ठीक है? और I'll ask you one more question. From one, दिव्यांशु 6, ओके, 3 नहीं 6, एक ग्लूकोज से. How many CO2 are released from one Krebs cycle, normal Krebs cycle? ग्लूकोज नहीं, नॉर्मल. इससे कितने CO2 निकल के बाहर आते हैं? कितने निकल के आते हैं स्टूडेंट्स? 2 CO2 निकल के आते हैं. 2 CO2 निकल के आते हैं. Okay. Now, can I ask you one different question now, students? Okay. Can I change the question? Question difficult करूं? How many CO2 are released? How many CO2 are released from one glucose in total? How many CO2 released in total? How many CO2 are released from one glucose in total? Okay. First things first, did I mention Krebs cycle anywhere? First things first, did I mention Krebs cycle anywhere? No. I didn't say Krebs cycle. I said in total. इसका मतलब क्या है? You should also consider link reaction. Also consider link reaction. Not tell me the answer. Not tell me the answer. Nice. Nice. 5 कैसे बनेगी स्टूडेंट्स? How is it 5? Okay, tell me students, the link reaction which I showed you, how many CO2 link reaction? How many CO2 students? How many CO2 link reaction? All of you. How many CO2 release in link reaction? कितने निकल के आते हैं link reaction के अंदर? 1 is for 1 pyruvate, है ना? 1 for 1 pyruvate. But what was my question? My question was from glucose. एक ग्लूकोज से 2 पायरुवेट बनते हैं. इसका मतलब लिंक रिएक्शन कितने बार हो रहा है? लिंक रिएक्शन कितने बार हो रहा है स्टूडेंट्स? 2 बार हो रहा है तब लिंक रिएक्शन, है ना? तो 2 CO2 निकल के आएंगे. 2 CO2 निकल के आएंगे एक ग्लूकोज से स्टार्टिंग में लिंक रिएक्शन के अंदर, और यहां पर कितने आएंगे? 4 आएंगे, राइट? So 4 plus 2 is 6, right? Students, do I need to explain again? Ask me again. Ask me again if I need to explain again. Okay? So please remember, एक ग्लूकोज है ना, एक ग्लूकोज से 2 पायरुवेट बनते हैं. 1 pyruvate here, 1 pyruvate here, है ना? This pyruvate undergoes link reaction. This pyruvate undergoes link reaction. Link reaction. यहां से एक CO2 निकल के आया, यहां से एक CO2 निकल के आया. This undergoes now Krebs cycle. Krebs cycle. इस Krebs cycle से कितने स्टूडेंट्स कितने निकल के आए? बताओ. 2 CO2 निकल के आए एक Krebs cycle से. 2 CO2 निकल के आए यहां पर एक Krebs cycle से. 2 plus 1 is 3, 2 plus 1 is 3, 3 plus 3 is 6. Clear? Hmm. 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 Okay. Now, students, now, do I need to make it more complex? I'm thinking, students, I'm thinking. Okay, I will make questions more difficult when I talk about this one, students. When we talk about the entire energy currency. Okay, energy currency. Okay. Okay, this is, oh, so do you realize they will not ask you the Krebs cycle? आपको Krebs cycle नहीं पूछेंगे आपको. They will only ask you questions like this. Okay. Okay. Tell me, students, how much of substrate level ATP is produced from from one glucose? How much? This is a very good question. On one glucose, how much substrate level ATP is produced? Oh God, all of you will hate me now. Substrate level. I'm asking substrate level. I am not asking you to include. I am not asking you to include this NADH, FADH. Pure substrate level. Let me think the answer, students. Let me think the answer first of all. कितने बनते हैं स्टूडेंट्स वहां पर चार निकल के आते हैं दो निकल के आते हैं okay I got the answer students correct all of you some are correct okay mm. Okay tell me students so students when I talk about substrate level how many do we get in glycolysis students how many do we get in glycolysis quick students quick when we talk about substrate level we should not include this we talk about substrate level right so इसका मतलब क्या है substrate level से कितने निकल के आ रहे हैं substrate level से glycolysis के अंदर 4 minus 2 is 2 ATP 2 ATP निकल के आ रहे हैं substrate level from glycolysis और हमारे Krebs cycles एक Krebs cycle से एक substrate level दो मतलब दो तो दो और दो चार substrate level है ना एक कैसे स्टूडेंट्स दो आएंगे Krebs cycle से I am talking about one glucose I am talking about one glucose here एक ग्लूकोज से एक निकल के आएगा दो से दो निकल के आएंगे है ना ओके क्लियर स्टूडेंट्स क्लियर गॉड मजा आ रहा है थोड़ा आज ओके आगे चले स्टूडेंट्स आगे चले क्या ओके दीज क्वेश्चंस आर वेरी इजी ओके स्टूडेंट्स आई एम ऑल गुड look at this question identify the steps in tricarboxylic cycle which does not involve oxidation of substrate they're asking you in which oxidation does not happen इसका मतलब क्या है they're asking you about substrate level कहां पर हो रहा है substrate level कहां पर हो रहा है स्टूडेंट्स बताओ sir how do we get two pyruvates from one glucose you need to understand for that my previous class everybody know students from one glucose how we get two pyruvate please watch my previous class glycolysis समझना पड़ेगा उसके लिए okay which where substrate level happens succinyl coenzyme to succinic acid यहां पर होता है substrate level है ना here substrate level happens here okay succinic acid to malic acid student देखो succinic acid to malic acid इन दोनों के बीच में FADH निकल के आ रहा है ना that is because of oxidation only है ना so that cannot be the answer isocyanated to alpha keto or normal malic acid to oxalic acid NADH normal oxidation है ना so this cannot be the answer so only here what happens oxidation does not happen only substrate level happens no, students क्या है students इतना easy questions you can't solve हाँ okay quick students I think we'll do a part 3 In part 3 I will do ETC. अगर ETC आपको पढ़ाना है and if you want to solve some good questions, we'll do a part 3. Okay, in part 3 I'll teach you ETC, mainly ETC. ठीक है, and I'll bring AIMS questions next class. I'll bring AIMS questions, I'll bring AIPMT questions in part 3. Okay, twice. बहुत easy question students, बहुत easy question है ये. NEET 2022 question. So this is why I tell you, NEET questions are actually easy if you solve them, if you write them. If you actually pay attention, NEET questions are very easy. Okay. Students, 5 carbon compound. 5 carbon compound कौन सा है? तो कितनी बार बताया आपको मैंने, यहां पर कितना, 4, 4 प्लस 2 6, 6, यहां पर 5, यहां पर आगे 4 आएगा, 4 आएगा, 4 4 4 4, राइट? क्या स्टूडेंट, कितना इजी क्वेश्चन है, कैसे कर सकते हो? नेक्स्ट क्वेश्चन, नेक्स्ट क्वेश्चन ऑन द बोर्ड. हाँ, students, again, NEET is not about lot about concepts, it's about accuracy and practice. NEET is all about lot of practice. You need dedicated practice for 1 year for NEET. Lot of PYQs you have to solve, a lot of questions, कुछ 1000+ क्वेश्चन सॉल्व करने पड़ेंगे आपको. Students, बहुत easy क्वेश्चन है ना? Students, this I'll teach you the next class. ठीक है? Yes. NEET is about data. NEET is about knowing lot of information and processing lot of information at once. क्योंकि आपके पास NEET is all about speed plus accuracy. वहां पर बहुत स्पीड होना चाहिए, आपको स्पीड होना चाहिए बहुत. ओके, आपको वहां पर ना 180 क्वेश्चंस 45 मिनट्स में सॉल्व करने हैं. नॉट 180 स्टूडेंट्स, लेट्स से हाँ, ऑलमोस्ट बहुत बहुत फास्ट सॉल्व करना पड़ेगा आपको. ओके, बहुत ही फास्ट सॉल्व करना पड़ेगा. दैट्स अबाउट नीट. Around 90 questions you have to solve. I'll tell you what, why NEET is difficult. Okay, 90 questions. Okay, let's say 180 questions in 3 hours. Can anyone tell me how many questions do we get per, how many questions per, right? How many questions do we get per this thing minute? Anyone? How many questions per minute do we get, students? Quickly in the chat. How many questions per minute? All of you. All of you. 3 hours is 180 minutes, right? Right. So let's say in this 3 hours, instead of 1 minute, let's say you get out of this 1 minute, आपको लगेंगे at least 10 seconds to read the question. So you're left with what? Level 50 seconds. 50 seconds. After that, you need at least 20 seconds to solve the question or 30 seconds to solve the question, है ना? So 50 से 30 seconds निकल गए आपके, 50 seconds से ना 30 seconds निकल गए वहां पर. So you're left with 20 seconds. 10 seconds will go to mark. So आपके पास actually 1 मिनट के अंदर ना 30 सेकंड तो ऐसे निकल जाते हैं. 30 सेकंड निकल जाते हैं सही. 10 सेकंड पढ़ने के लिए, 10 सेकंड पढ़ने के लिए और 10 सेकंड्स लिखने के लिए. तो 20 सेकंड निकल जाते हैं सही. 20 सेकंड ऐसे निकल जाते हैं, है ना? सो आपको एक्चुअली 180 क्वेश्चंस, है ना, पर क्वेश्चन यू विल गेट अराउंड 40 सेकंड्स. 40 सेकंड्स पर क्वेश्चन. और अगर सोचो अगर आपको उस 40 सेकंड्स में एक क्वेश्चन को पूरा सॉल्व करना है, अगर आपको एक लेट्स से बायो में तो हो जाएगा कभी कभी, बट अभी क्या हो रहा है पता है, बायो में आपको क्वेश्चन पढ़ने के लिए वी आर टेकिंग 20 सेकंड्स, क्योंकि स्टेटमेंट बेस्ड क्वेश्चन ज्यादा आ रहे हैं. सो यू एक्चुअली गेट ऑन एवरेज 30 सेकंड्स पर क्वेश्चन, और आपको ये करना है ऑलमोस्ट फॉर 3 आवर्स कंटिन्यूअसली विदाउट टेकिंग अ ब्रेक. Without taking a break, directly you have to start every 30 seconds, question solve, question solve, question solve, question solve, exactly for 3 hours you're supposed to do. Okay, to get full marks, that is, to get full marks. Okay, right. So that's the difficult, students. Mm. Okay, students, Divanshu, I have taken diversity in full before. No, I have done living world, I have done biology classification, both are done before, right? Okay. So NEET is all about accuracy and how fast you can think, how fast you can process the answer. Okay. And imagine solving physics numericals which are JEE mains level, right? Which are, can you do them in 30 seconds? Can you do physics numericals in 30 seconds? Fast numericals. बहुत difficult, student, बहुत difficult होगा. So that is about NEET. It questions are easy little, but fast answer should come. So NEET is all about discipline and remembering a lot at once. Okay. So thank you so much, students. Bye bye. Take care, all of you. Right. So thank you so much, students. Bye bye. I'll see you next class. I'll try to bring more difficult questions from next time. Right. I'll bring AIMS and AIPMT questions. So let's solve them and have fun. Okay. So bye bye, all of you, students. Bye bye. Students, see, an accelerated, I will try to look for most chapters which I have not done for faster revision. है ना? But chapters which I've, if I do all the chapters which I've already done before, what happens is sometimes you will not get, I will not be able to cover the chapter syllabus then. That's the problem. Okay. Again, exam may be difficult, but NEET exam selection ratio is the toughest. It is more toughest. It is the toughest actually. The selection ratio in NEET examination is less than 1%. Friends, do you know that? The selection ratio of you getting a government medical college is less than 1% in the entire world population. If you take also like the exam who give people who give the exam, less than 1% is selection ratio, right? So bye bye, students. Bye bye. Yes, I know for SGS also, but NEET is around 0 point something, 0.7 something. Okay, so bye bye, students. Bye bye. But at least competition is low for all of you. But students, do you realize there is an age limit for SGS? There's the age limit. Okay, so when there is an age limit, you will feel relaxed. पर NEET के अंदर ना 5 साल 6 साल पढ़ते रहते हैं. You compete with people who have been studying for 3 years, 7 years, 8 years more than you. So bye bye, students. Bye bye.